is not 100% accurate. I try to make these videos as accurate as I possibly can, but there's always a chance that I'm remembering something incorrectly or learned something incorrectly, so I'm sorry if there's anything that isn't 100% correct, but I'm trying my best. So I hope that you're able to sit back and enjoy this, and I will again try to put like pictures or diagrams to help me explain things on the screen, just because I think it's easier if you can actually see the concepts. Maybe soon in the future I'll try to do a video again where I actually like draw out the concepts and write stuff down, but those are kind of harder to film, and I liked doing it like this last time, so let me know which way you like better. I don't know, but I will try to put some diagrams and things on the screen. But let's get started. So the first big concept that I'm going to teach you about is chromosomes and chromosomal abnormalities. So you may know that for the vast majority of humans, you have 46 chromosomes in every cell of your body, and there are 23 unique chromosomes, and you have 2 copies of each of those 23 chromosomes. You get one from your mother and one from your father, and that makes 46. 2 times 23. So chromosomes 1 through 22 are known as autosomes, and all males and females get one copy of each of those from their mother and their father. And then there's one sex chromosome where females get an X chromosome from each of their parents, whereas males get an X and a Y chromosome. So that's the only like chromosomal difference between biological males and females. And so, like I said, you get one copy of each of those from your mother and father, and that happens when a sperm cell and an egg cell fuse to make a zygote. So I'm going to talk about how you get those sperm and egg cells that only have 23 chromosomes rather than 46, so that when they fuse, you get 46 chromosomes in the baby. So what happens is a process called meiosis or meiosis. I never knew how to say that. I don't know. But essentially there are cells called germ cells, and they are destined for reproduction. And so what happens in those is basically they have 46 chromosomes, but they go through a process where each of those pairs of chromosomes, so like chromosomes number 1 and chromosomes number 2, they all line up in the cell, and they are separated into 2 separate gametes, which can be either egg cells in females or sperm cells in males. So ideally, each sperm and egg cell would get the appropriate 23 chromosomes, one of 1 through 22 and one sex chromosome, so that when that gamete fuses with another gamete, you get the appropriate 46 chromosomes. However, meiosis doesn't always go the way that it is supposed to. There is something called Nondisjunction, which is basically where the chromosomes don't separate properly. And so sometimes what happens is one of the gametes, let's say an egg cell, gets 2 of one of the chromosomes, and so the other gamete would only get none. It wouldn't get any of that specific chromosome. But when one of those fuses with a sperm, you're not going to have that 46 chromosomes. So if it is the egg cell that gets 2 of one of the chromosomes, and then it fuses with a sperm that got the normal 1 chromosome, zygote has 3 of that specific chromosome, and that has a lot of impact on the zygote, because having 47 rather than 46 or 45 rather than 46 chromosomes, it's going to have a huge genetic impact on that child. And unfortunately, the vast majority of the time that this non-disjunction occurs, it ends in a miscarriage, because most of these chromosomal abnormalities are not compatible with life. You can't really live with that big of a difference in your genetic material. However, you have most likely heard of Down syndrome, and maybe you've also heard it called trisomy 21. So what a trisomy is, it basically means 3 chromosomes. So it's what I just explained, that's what happens. But in trisomy 21, you have 3 of the 21st chromosome specifically. So in one of the parents, one of the gametes got 2 of that 21st chromosome, and when it fused with the gamete that had 1, you get a zygote that has 3 of the 21st chromosome. There are actually other ways that it can happen, but that is the most common way to end up with 3 of the 21st chromosome. And obviously, you probably know people with Down syndrome, so it's definitely livable. It is something that can be lived with, and the reason for that is that the 21st chromosome is actually the smallest chromosome out of all of them. It doesn't really have a whole lot of genes on it, so it kind of has the least impact, or the lowest percentage of genes are on that chromosome. And so that is why when you end up with 3 of it, it doesn't have as big of an impact as, say, having 3 of the first or the second chromosome, which are the largest ones. And so that's why you are able to live with it. However, it might be kind of interesting to know that all other trisomies or monosomies, which is where you only have 1 of a chromosome, are really just as common to happen in gametes. It's just as common to make a zygote with that number of chromosomes. It's just that they're not compatible with life, and so that's why we only really see Down syndrome or trisomy 21, is because the 21st chromosome is the smallest one. Now, the others that sometimes occur are trisomy 18, so 3 of the 18th chromosome, or trisomy 13. Those happen sometimes. They're called Edward syndrome and Patau syndrome, I believe. Those occasionally occur. A lot of times they do end up in miscarriage. Sometimes the babies are born. Typically, if they are born, they don't live past like a year. So they definitely have a larger impact, but those two do happen sometimes. You can also have chromosomal abnormalities when it comes to the sex chromosomes. So, like I said, normally you end up with 2 X chromosomes, or an X and a Y, but that same non-disjunction can happen with the sex chromosomes. And so actually the only monosomy that is actually compatible with life is the words that they often use in the genetics community, saying something is compatible with life, is having only 1 X chromosome, and that's your only sex chromosome is 1X, and that is called Turner syndrome. And it is definitely livable, compatible with life. These people are born females, but I believe they're infertile and have quite a few other genetic issues, but it is the only monosomy that's actually able to be born. There's also something called Klein meiosis, essentially male and female is determined by the presence or absence of the Y chromosome. And so even though they have two X's, because they have a Y, they're going to develop into a male. And when I talk about sex in my genetics videos, I am only talking about genetic sex. I understand that that does not have to do with gender identity. I'm not talking about gender identity at all. I had someone comment that on one of my videos asking like how I felt about gender identity and genetics. Totally separate. You are totally free to identify however you identify. However, sex chromosomes are what determine your biological sex, and nothing can change that. No matter how you identify or the hormone replacements or surgeries or anything, like it's not going to change those sex chromosomes. So when I say female or male, I'm talking about sex chromosomes, but that's my little like disclaimer. But yeah, Klinefelter syndrome and Turner syndrome, I believe, are the most common aneuploidies when it comes to sex chromosomes. I don't know if I said the word aneuploidy, that's basically a word meaning any difference outside of the normal when it comes to chromosome number. But yeah, so that was kind of my fun fact that yes, Down syndrome happens, but it's just as likely for non-disjunction and trisomies and monosomies to happen on all other chromosomes. It's just that they are not compatible with life. And the other thing going along with that is that the vast majority of the time, those issues with the number of chromosomes come from the mother. Most of the time it is the mother that leads to that, and maternal age is one of the biggest factors in determining if a baby is going to have a chromosomal abnormality. And that is because females are born with all of the eggs that they're ever going to have, all of the egg cells that they're ever going to have. But basically those cells are stuck in the very first phase of meiosis until the point where they are actually released. Like, I guess the point where they actually fuse with a sperm. And so as the mother ages, her eggs are getting older and older and older, and once they do actually go through meiosis and separate, the older she gets, the more likely it is that the chromosomes aren't going to separate properly, and you're going to get two or none of a chromosome, and thus end up with a trisomy or monosomy, specifically something like Down syndrome. So yeah, my parents were rather old when they had me. My mother was 43, and so I think she had like a 1 in 30 chance of me having Down syndrome. But very lucky that I do not have Down syndrome. But yeah, maternal age is one of the biggest factors that leads to trisomies and monosomies. So the next thing that I'm going to talk about when it comes to chromosomes and meiosis is something called genetic linkage. And this is something that wasn't really discovered by Gregor Mendel, who is the father of genetics. He basically discovered all of genetics. But kind of a fun fact about that is that he did all of his research in pea plants, and that was how he came up with like recessive and dominant genes. I talk more about that in my other videos. But he originally wanted to do his research in mice, but because he was a monk, it was deemed that that would be inappropriate for him to study mice because mice have sex. And so he was forced to do his research in pea plants rather than in mice. And it's kind of funny because if he had studied mice, he probably would not have come to the conclusions that he did, and maybe wouldn't have, like, discovered genetics, because the genetics of mice are so much more complicated than that of pea plants, and so he probably wouldn't have seen the super clear-cut percentages and traits that he saw in the pea plants in mice. You know, like, imagine trying to study genetics in humans by just looking at how they look. It'd be kind of hard to do that because we don't always get like a perfect mix of our parents. So pea plants were very simple, and that was kind of why he discovered genetics. But anyway, my little fun fact tangent. So he came up with what is called the law of independent assortment, which, like I said a couple minutes ago, when all of those chromosomes separate, he was basically saying you are equally likely to get any combination of genes from your parents because those chromosomes separate independently from each other. So, like, you get one of your mother's chromosome 1, and that has nothing to do with which of your mother's chromosome 5 you get. So, independent assortment. Basically saying, like, if you are a green pea plant, that has nothing to do with if you are a tall or a short pea plant. But genetic linkage kind of throws a wrench in that. And so that is the concept where genes that are located on the same chromosome are much more likely to be inherited together than genes that are on different chromosomes. And so I think this was actually discovered in fruit flies, I think. But basically, there are certain genes that are located on the same chromosome that you're more likely to inherit those traits together. And an example of that is hair color and eye color. So if you have brown hair, you're much more likely to have brown eyes. And I think the same goes for blonde hair and blue eyes, and red hair and green eyes. And so in meiosis, like I said, before those chromosomes separate, what happens is something called crossing over. And so basically, like, your mother's two chromosome 1s come together in the middle of the cell, and they exchange genetic material with each other, and then they separate. So that's kind of how you get so much variation, because obviously if that didn't happen, there would be, like, your parents wouldn't have that many possible combinations that they could make in babies. But obviously we know that they're not gonna give birth to, like, the same kid twice, you know, unless you're identical twins. So that crossing over and the sharing of genetic information is one thing that leads to a lot of variation in the kids that your parents could make. But so basically, it's kind of hard to explain. I'll try and put, like, a graphic here so that it makes more sense. But the closer that genes are located to each other on a chromosome, the less likely they are to be separated from each other when those chromosomes cross over. And so, like, if two genes are located right here, you're most likely to get the exact same two traits as the parent that gave it to you, because they're not going to be separated from each other. I don't know if that really makes sense, but my best example of that is, like, red hair and green eyes and so on. And in doing that research, I found out that red hair and blue eyes is, like, the least common combination ever. Like, it's super rare. So if you have red hair and blue eyes, I'd love to hear about it, because that's very interesting that you ended up with that. But I have probably the most common combination ever, which is brown hair and brown surprise, surprise. But yeah, I think that is all that I was going to talk about when it comes to chromosomes. I hope that made sense. I feel like it was very incoherent, but you guys always say that when I explain genetics, it makes more sense. I hope it did. But yeah, that's what I wanted to say about chromosomes. And the next big topic that I'm going to talk about is mutations. So we talked about chromosomal abnormalities. That's one cause of genetic inherited diseases. Another thing is mutations, where the specific sequence of your DNA is altered rather than just the number of chromosomes or the type of chromosomes. You probably know that DNA is basically the code that makes you who you are. So you have over 3 billion what are called nucleotides in your DNA, and it's made up of G's, T's, C's, and A's. That's like the letters that make up the book of you, the book of your DNA. And those are adenine, thymine, guanine, and cytosine. Yeah. And so that whatever sequence of A, T, C, and G that you have codes for you. But how does that happen? So I'm kind of gonna skip through RNA because I feel like it's not extremely necessary to like the high level explanation of how this happens, but basically your DNA is just the recipe book. It's just the code for making you, but it doesn't actually make up you. What makes up you is proteins. So the DNA codes for the proteins that are going to be made, and those proteins are the things that do all of the things in your body that make you have blue eyes and make your heart pump, and everything that happens is done by the proteins, which are made based on what the DNA says. So basically, each 3 nucleotides in your DNA, A, T, G, C, T, A, G, T, C, whatever, each 3 codes for a specific amino acid. And amino acids are the building blocks for protein. Building blocks for proteins. So each protein is going to be a string of amino acids that are each coded for by that DNA sequence, by each 3 nucleotides. And when you have just the slightest mutation, like changing a T to a C or whatever, it's going to code for a different amino acid. I'll try and put the diagram here, but basically every combination of 3 nucleotides that you can think of codes for an amino acid. And so when you change one, it might code for a different amino acid, and it's going to make a different protein, and that can cause all of the issues that you see with different diseases. So the first type of mutations is called silent mutations, and you might be able to tell from the diagram that I'm going to put on the screen that there are different codons or sections of 3 nucleotides that code for the same amino acid. And so in some cases, a nucleotide will be changed. It's a mutation, but it still codes for the same amino acid, and so you still get the same protein. And so you don't see any impact. It doesn't really matter that there's a mutation. Now, a missense mutation is a mutation that occurs when that actually changes the amino acid. So if you get a specific nucleotide change that causes a different amino acid to be made, that's a missense mutation, and that's when you start to get issues. You start to get a different protein. The next thing is nonsense mutations. That's what they're called. So from the is a sequence of 3 nucleotides that triggers the end of the protein. So you have a whole like sequence of amino acids, and then there's nucleotides that say, hey, we're done with the protein. And so that protein is finished. It goes on to do its protein things, whatever its role is, because of that stop codon. Well, sometimes when you get a nucleotide change in that sequence, it goes from, say we're coding for one amino acid, but now because of that different nucleotide, now it's a stop codon. And so we stop making the protein prematurely when we're supposed to have 10 more amino acids, and we just stop. So that's pretty bad when you stop prematurely, and oftentimes that results in like no viable protein, and you get a pretty severe disease. So missense mutations are going to occur when you just have the change of a nucleotide, but something else that can happen is taking away or adding in a nucleotide, so insertions or deletions, and those can be a lot more impactful because try and think of it like reading a sentence. If you take out one full word of a sentence and you shift everything else over, the sentence is probably still going to make sense. You're still gonna make the protein. It's kind of like taking out one ingredient of a recipe. You're probably still gonna, for the most part, get a similar cake, a similar baked good. So that's what happens when you have a missense mutation. But let's say we just took out one letter of a sentence, and then we shoved everything over and put everything back together. Well, now every single word after that letter that you took out is going to be jumbled. Every word is now different. Every ingredient is now different. And so you are going to have a vastly different protein, a vastly different recipe, because all of that is going to be shifted over, and every codon thereafter is going to be different than what it was supposed to be. And so that often results in a nonsense mutation, because if you're changing all of the codons, you're probably going to get one that codes for that stop codon and is going to cause the end of the protein, and you're not going to get a good protein. And so something that I was going to discuss is muscular dystrophy. This is a disorder that is caused by these types of mutations, and it's something interesting. So muscular dystrophy is actually a sex-linked disorder, meaning that the gene that codes for the dystrophin protein is located on the X chromosome. And like I said, females have 2 X's, males have 1, and I explained all about X-linked traits in my last genetics video, so you can go watch that. But essentially it means that males are much more likely to have the disorder, so males are more likely to have muscular dystrophy. But that's not really the point of this. Dystrophin, the gene that codes for dystrophin or dystrophin, is the largest gene in the body. It has the most genetic material. I haven't really told you about exons, but the most exons of any gene. Maybe I'll talk about that in a future video. And that dystrophin protein is very, very important for your muscles. And so there are 2 main types of muscular dystrophy. One is Duchenne muscular dystrophy, and the other is Becker muscular dystrophy. And so the difference between acids, but if you add in 1 or 4 or 7, you're now not going to have a multiple of 3. It's going to shift the whole reading frame of the gene, and a lot of times what happens is you get that nonsense mutation, you get a stop codon, and what happens is you don't end up with any viable dystrophin in the body, and you get a very, very severe form of muscular dystrophy. However, Becker muscular dystrophy happens when you have an in-frame mutation, so you can have an insertion or a deletion in that gene that codes for dystrophin, but outside of that, where you have a mutation and you're probably going to get some mutated amino acids, outside of that, the rest of the dystrophin protein is going to end up relatively normal, and so you get a much less severe form of muscular dystrophy. So that is my big example of like how frame shift mutations work, and I hope I did an okay job of explaining how transcription and translation works, how proteins are made from DNA. Maybe I'll try and do a better explanation of that in a future video, but essentially your DNA is the recipe. It has 3-letter words, all 3-letter words, and each of those is a different ingredient, a different amino acid, and that makes a protein, which is the final cake. And when you have any issue in your DNA, any change, it can lead to a different protein, which can lead to disease. So, yeah. One mutation that I'm going to talk about, this is just a little fun fact that I learned in my last genetics class in college, is in a protein called CCR5. So CCR5 is actually the receptor on your cells that is responsible for letting HIV into your cells. You probably know what HIV is. Very deadly virus, sexually transmitted disease, and in order for a disease to impact you, or a virus to impact you, the virus has to be able to get into your cells, and CCR5 is a receptor that allows HIV to bind and get into your cells. But if CCR5 is mutated, HIV can't bind, and so HIV can't get into your cells. And so this is an example of a mutation that leads to a defective protein, but where the impact is actually good for the person that has the mutation. Usually we think of mutations as causing bad things, causing you to be sick and have disorders, but if HIV can't bind, you can't get HIV, and so you might not know that there are people that are completely resistant to HIV if they have this. It's called a delta 32 mutation in CCR5 that makes it where you cannot get HIV, and it's actually only like 1% of people that are fully homozygous for that mutation, meaning that they literally can't get HIV. I think it's mostly in European populations, and then it's another like 10 or 15% of people that have one copy of that mutation, and so they, I think they're more resistant to HIV, but they can still get it. But yeah, just an example of a mutation that causes something good in the person. To end on a slightly lighter note than talking about Down syndrome and chromosomal abnormalities and muscular dystrophy. But yeah, so this video was all about chromosomes and mutations. Mostly kind of a bad video, but teaching you about how all of that works and how you get answer any of your questions. Yeah, I hope this made sense. Again, it's really hard to teach things and